All right, so check your scores. Excuse me. For your lab practicals, I finished grading them, and the scores have been posted. Remember, this is out of 150 points. We can certainly go over them too. So if you want to talk about it in class next Monday, let me know. Excuse me a second. Seems like sometimes just as I start, my throat starts to get a tickle in it. Anyway, so we can go over your lab practicals, no problem at all. We can talk about what you did right, what maybe you didn't do right. Do keep it in perspective again, though. There's still a lot of points left in the semester, so if you didn't do as well as you wanted to do or thought you might have done, then there's no sense for despair. We still have the field notebook and the lab practical, many other assignments, so obviously you'll want to learn from your mistakes. So if you look at your lab practical and you don't understand why you got something wrong or where you went wrong, then by all means, let's talk about it. But in the end, it is 150 points out of however many there are in the class. There's, what, over 1,000, I'm sure, but I don't remember exactly how many there are. So just keep that all in perspective. Of course, you'll want to do better if you didn't do as well as you thought. But then again, or not then again, but that's where you'll want to talk to me about that so we can figure out how maybe you can do better. All right, so the last time we got together in Chapter 3, we were talking about flowers, not about plants. We were talking about flowers among plants, or the types of plants that make flowers. And this was the last group of flowers that we're going to discuss. And as I mentioned in this group, it really is all about the flowers. It's all about the flower color, the flower shape. But it's all about the flowers in the sense that what the flowers are doing is attempting to attract something. They're attempting to attract pollinators. Because if you remember, once again, of course, back going to Bio 1, one of the most important characteristics of all living organisms, characteristic that all living organisms have to have, is the ability to reproduce. So however you go about that, those who are most successful at doing that, and since over 200,000 species of flowering plants, isn't that what I said? 250,000 species of flowering plants. Quite clearly, these plants with the flowers have figured out that this is a really good way to reproduce and perpetuate the species. So we were focusing our attention on that when we finished up the other day, about the different colors, whether in visible light or in natural light. Remember, once again, I also told you that oftentimes we can tell something about the reproductive nature of a plant by its flowers, in the sense of who the pollinators are, like hibiscus. Butterflies are very attracted to oranges and to yellows and to reds. Those plants with smaller flowers, I think I brought the example of a grass, right? Those plants with smaller flowers or less conspicuous flowers are going to be less likely to want to attract pollinators, or less likely to evolve to attract pollinators, but be more wind pollinators, right? So think about all the energy that living organisms take in each day and what they do with that. Think about that as a budget, so that you have certain things you have to take care of each day. And throughout the life of an organism, reproduction is one of those things you have to budget energy for, right? Whether male or female or otherwise, there is an energy expenditure associated with it. So if you're not going to worry about attracting pollinators, don't make really big fancy flowers. That's a lot of energy wasted. Make small flowers and let the wind do the work, right? If you're going to do the pollination thing, though, with insects or some other type of pollinator, then you're going to want to maybe invest a little more, if you will, of that energy into these attractive qualities that flowers have for those pollinators, right? It all fits together. Whether you're attracting them because they get a meal, whether you're attracting them because you're fooling them into thinking they're a mate. But what do you want to do? Is you want to crawl in there and get that nectar, and you, at the time or when that occurs, as I showed you in this flower, that stamen, and the stamen, of course, remember, is where the anthers are, and that's where the pollen is. So all that part of the top and the yellow is pollen is going to be dusted all over that insect, and when he moves to the next flower, if it's one of those flowers that has both sex organs, which is the most common type of flower. I mentioned the other day there are some that are only male and female, but most flowers have both. But in the next flower, that pollen is going to be used to pollinate the carpel, or the female part of that next flower. Now, speaking of the carpel, that female part of the flower, the carpel, as a lot of things are in plants, among flowering plants, is a modified leaf. Remember, the cone of a cone-bearing plant was a modified was a modified shoot or modified part of the basic structure of a plant. So once plants develop these broader leaves, like we talked about back in the earlier chapter, then we can imagine, if we look at something that we're really familiar with today, green beans, we can imagine a scenario whereby, like, on the leaves of some ancient plant, right, some ancestral plant, that these ovules, or remember, these seeds, begin to develop. And that if you protected your offspring, as I talked about the other day, and we've mentioned, this semester, if you protect your offspring, then you got a better chance of them surviving. And by doing that, you also can invest less in reproduction, right? Remember, we said there's on the two extremes of reproduction, or make lots of offspring, because most of them don't survive, because you provide little, if any, parental care, right? Or you have fewer offspring because you're going to provide some parental care in the hopes that most of them are going to survive, right? So it's a kind of, and organisms fit somewhere in between. So imagine some ancestral plant leaf with these ovules that, through mutation, through sexual recombination, through all those mechanisms that we talked about early in the semester, develop this ability to protect, to fold over on those ovules. That would be selected for, wouldn't it, right? Mother Nature would select for that because that would increase reproductive rate. And remember, the bottom line, always remember, if you think about when you think about an adaptation of an organism today, and you have to think of it in the idea, how does that adaptation allow that organism to have an advantage in its environment, right? So this would be an advantage. You're protecting your offspring. And so we see it in our modern day green beans. Inside of the green bean, the little bean part inside, that's the ovule, that's the seed. And the part on the outside, the green part, right? The green pod, right? Pod, that's what they were called. That's the carpel, right? That's the fruit. That carpel is the, oh gosh, let me go back to the anatomy. Sometimes when we see it all modified like that, we lose track. Where it's this thing, right? The carpel, right there. It's that part that's got the stigma, the style, the ovary, the ovules. Hi, Jesse, how are you? The seeds. Oh, there's those those flowers that are male and female. Right? So that's what that is. And inside of the carpel, great. Inside of the carpel are the ovules, the seeds. Now, we come along and we eat green beans, and so, like we discussed the other day, things with seeds are fruit. So green beans, you might call them vegetables in the common name kind of sense, but they are fruits, right? They have seeds in them. And what you're eating is the carpel. You're eating the fruit. That whole vase-like structure, which looks an awful lot like an archegonia, doesn't it, huh? Back in the mosses, that whole vase-like structure is the carpel. All right. And that carpel can become all kinds of different fruits. And there are fruits that are tomatoes, grapefruits, nectarines. There are fruits that are fleshy, like peaches. Nah, real crazy about peaches because of the fleshiness. Mangoes. My wife loves mangoes. I like the flavor of mangoes, but the fleshiness of the mangoes, right? So there are fleshy fruits. There are juicy fruits, kind of like the gum. There are dry fruits. That's what nuts are, hazelnuts, milkweed. That's a fruit. Anything that surrounds those of seeds, anything that is that thickening of the wall of the carpel, that is a fruit, and its job is to protect the seeds, right? Because you're surrounding it. Things like desiccation, a nice juicy, like a grapefruit, like in that picture, would protect those seeds from drying out. Because, man, I love grapefruits, and you know why? Because they're real juicy, right? And tomatoes are that way. In the nectarine, what they've done is take part of the carpel and turn it into this really hard pit thing, right? In nectarines and in peaches, avocados have got them too. That really hard part in the middle of the pit is protecting the seed. So it can protect the seed, and what it also does is if you make it real edible, then it aids in dispersal. Look at the nectarine, right? Something munching on a nectarine in the wild would certainly be able to probably chew through and digest this, but man, that ain't going to digest. That's going to come out the same way it went in, right? But meanwhile, while it was doing that, while it was making its way through the process of the digestive system of the critter, that critter has moved. So dispersal has happened, right? Oh gosh, the other day, remember I told you that there are some seeds of plants that won't germinate until they've been roughed up by acid, like stomach acid, passing through the digestive tract of a critter. One of the things that we learned as we kind of go further on in this class, at least I hope that kind of is starting to come into focus, is this idea of, I don't know what you want to call it, mega evolution, where like evolution occurs among individuals, or among populations of individuals, right? But it also evolution occurs at much higher levels between organisms and interactions between organisms, right? So this whole idea of fruits and what fruit is all about and what it does is one of those mega evolution things, right? It's making other critters want to do your bidding. Your spreading of the seed out there, right? By making it advantageous for you to want to come and mess with me, right? To maybe chew through that tomato, dropping seeds on the ground or passing through the digestive tract. So there, and just kind of point out the different types of fruits. Don't get too freaked out about, oh my gosh, is he expecting me going to know the difference between a true berry and a drape and an aggregate fruit? But no. But like we talked about the drupes, those are the ones with the hard pits and what that's all about. See what's called the pericarp and the exocarp and the mesocarp, endocarp. Those are all parts of the carpel. So the outer part is the exocarp and the middle part is the mesocarp. So when you eat a nectarine or a peach, you're eating mesocarp, and the pit is the endocarp and the seed is inside of there. See how well that's protected? Raspberries, blackberries, what they are is aggregate fruit. So what an aggregate fruit is, it's one flower that has many, many ovaries in it. So it's got many carpels. Okay, so it'll have perhaps many anthers too, but a lot of times flowers will have one or just a few carpels, female parts. So they'll have maybe several pollen anthers, but just a few of the carpels. But in these aggregate fruits, that's where the aggregate, we say that the aggregate part is there's a collection, many carpels. And so each one of those little, oh gosh, if you think about these, oh, especially blackberries, right? When you munch up, each one of those little balls of fruit juice that you munch through that is so good, each one of those represents a fruit, right? So it's a collection of fruits. Legumes are the beans. Those are fruits. Legumes are real important plants because they feed a lot of us, and they're also important plants because they're nitrogen fixing capabilities. Remember, they were the ones in, oh gosh, back in the prokaryotes that have the nodules growing on their roots, where the nitrogen-fixing bacteria live, and legumes are known to have or be high concentrations of having those nodules. Samaras, which I think is a really cool name to be, is if you look at it at first glance, it's kind of confusing, right? It looks a little bit like a, well, hopefully it's not confusing. It looks a little bit like a pine seed, the one I showed you yesterday, right? Now, the pine seed, of course, is not part of a fruiting plant. So this is a really cool example of convergent evolution again. I'm probably thinking, how many times is he going to say that this semester? But notice what Mother Nature does to critters, right? So these critters aren't going to use pollinators or insect. I'm sorry, not pollinators. These organisms are not going to use things eating them to disperse them. They're going to use wind, right? The pericarp. The pericarp, again, the carpel, the outside of the carpel, the outside of the fruit, if you will, gets modified into a wing. The seed is contained in that little tiny protected area in the corner, and the wind takes it off. So these plants, like maples and elms, red maples here in Florida, they're going to go with the wind. They're going to go with the wind as a dispersal for fruit. Now, don't confuse, insect pollinators, critters that move pollen around, and wind can move pollen. Critters can move fruits around, and wind can move fruits around as well. Okay? Right, so fruits, so pollen, then fertilization, then you get a fruit. Remember where this all is in the life. Pineapples. I love pineapple too. Pineapple is kind of like an aggregate fruit, but it's an aggregate flower, or what they say in this term, multiple fruits. What we basically have here is each one of those. If you look at it like on the right, and when the pineapple is sliced down the middle, each one of those sections, you can kind of see it in the picture. Each one of those is a flower. Pineapple flowers. If you look at pineapples online, really, really pretty. Pineapple flowers grow out of here. Each one of these little things is a flower. So in a pineapple, what you're eating, right? What you're eating is the fruits of many flowers. Unlike the berries up above, which is one flower, right? One flower, lots of carpels. Pineapple is many, many, many flowers, right? Each one of those is circle is a pericarp of a flower, is an individual female part of a flower. Look up pineapple flowers online. They're pretty. Oh, and of course, like I said, right, what's this all about? Dispersal, dispersal, dispersal. Wind, explosive action, witch hazel, one of the ones that shoot them out, kind of disperse. Critters, you know, edible berries, and things that stick to other critters. So in a barb, like an organism like a beggar tick, the beggar tick thing, the thing that sticks to you is basically a fruit. Is what it is, right? It's modified, so those kind of organisms, those kind of plants, they're not going to spend a lot of energy in making a nice juicy mesocarp like the peach does, right? Because they're not really going to be into you eating them to be dispersed. They're going to stick to you. But that's still a fruit, is what that is. It's just been modified, or parts of the fruit can be modified into wings. Again, explosive action can be used for dispersal. So you would expect a seed that comes from something that uses explosive action to not have a lot of edible material in it, right? Because that's not what it's using. Of these, the ones with the most edible material would be the ones that mouse is chewing on, right? Chew on me, take me up in a tree, be sloppy, drop seeds everywhere, blah, 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 poop me out somewhere else, right? And you're dispersing what I want. You're moving me around. Herbivore cache. That's what these little cache. That's what these rodents tend to do. Is they make these big caches, these collections of food. And oftentimes, what they do is they move berries around and seeds of other plants around. So they're very much, like I said, involved in dispersal. Then there's the wind. Coconuts can float, like you see in the picture down below. Mangroves. That's what they do. You know, they float. And all of these ways, these mega evolution ways that I've been talking about, and using the wind and all of that stuff together, that's what allowed, when you think about plants, who the dominant plants are out there, 250,000 different species of flowering plants. It's these guys. It's all been about the flowers and about the fruits. So let's kind of dig into a little bit closer these flowering plants. Let's take a look at the whole idea of how they reproduce here, because this is interesting. It's all about taking care of your offspring. So let's start with the flower up above in the upper left. And a flower has a female part, the carpel, the male part, the anther. Right, the stigma is the anther and the filament together. I know those terms sometimes get a little familiar what you're talking about, so you got to make sure that you really kind of take a look at them. So the carpel is going to be the stigma at the top, the style, which is what the carpel in the label is pointing to. Then down below is the ovary inside of the seeds. But so what basically a fruit like a tomato does is the carpel, the style and the stigma fall off after the thing becomes fertilized. So what we have is a sporophyte plant, a flower on a spot. So we're keeping with the same terms in the alternation of generation, and it's diploid. Let's go with the anther first. So during the process of meiosis, on the tips of the stigma, where the anthers are, and the tips of the filaments are, there are microsporangium. Small, male, sporangium, spore-producing structures, right? And these microsporangium produce microsporocytes. Now, microsporocytes divide by meiosis for variability. Remember, that's why meiosis does, and makes microspores. All right, now that microspore then divides through mitosis, and what you end up getting is a thing called a pollen grain. Inside of the pollen grain is the male gametophyte. The male gametophyte, the things we looked at under the microscope with the mosses and the fern, and in the male cones, we haven't looked at those under the microscope yet, but we looked at them in class the other day. That's where that's where gametophytes have gone, folks. They've shrank, got smaller and smaller and smaller. So now the male gametophyte is not that thing that I said it looked like soccer balls when we were looking on the gametophyte for the fern, didn't I? All right, looked like little soccer balls. That thing has shrunk down to the size that it's only just a few cells encased in pollen. Now, by the way, or encased in a pollen grain. That pollen grain around the outside has sporopollenin. That stuff you got to boil in acid, remember? All right, so it's very durable. All right, now let's jump over to the female side because on the right they're beginning to come together, and I want to get the female part first. So back over to the carpel, coming around through meiosis, or excuse me, in the carpel, in the ovary, in the ovule, there's a megasporangium, right? And megasporangium produce megaspores, and megaspores through meiosis produce, megasporangium produce megaspores through meiosis. And now remember, let's do a little bit of numbers here. When a megaspore undergoes meiosis, it's going to produce 4 surviving megaspores. Anytime a cell undergoes meiosis, it produces 4, right? That's back in Bio 1. So that's where the 4, if you see in the picture, the 4 little purple dots. Only one of those, that's why it's called surviving, is going to survive. So a megaspore is going to divide through meiosis, produce 4 megaspores. Right, they're still called megaspores, right? 3 of those are going to die off. One's going to then divide through mitosis 3 times and produce 8 cells. 8 cells. Now, that picture that follows right down below, I'm going to blow that up in just a second so we can see a little more detail. But those 8 cells are going to become things called antipodal cells, polar nuclei, synergid cells, and then, of course, got the egg, right? This is where the female gametophyte has begun. The female gametophyte, that thing that looked like the vase, right, has shrunk down and shrunk down and shrunk down and shrunk down to the point where now it's nothing more than just these 8 cells enclosed in something called an embryo sac. And then each one of those things that you see on the right, now we put the 2 pictures of the male and female together, each one of these little things down here that you're looking at is one of these. And inside of that, there are female gametophytes. And pollen, which contain male gametophytes, stick to the stigma, and then a pollen tube. You see back in the pollen grain, there's something called a tube nucleus. It's a cell, and a tube nucleus creates a pollen tube. See, it's leading the way. Come on, guys, go this way, right? Right behind it, there are 2 sperm. You'll see in a bit why there are 2 sperm. That's unique. That's interesting. And the tube nucleus is forming the pollen tube, and the sperm are following the pollen tube. And they come around and they turn the corner. And I'm going to jump off the slide for a second. Am I? Yes, I'm going to jump off the slide for a second because when they come around and they turn the corner, those 2 sperm enter into the embryo sac through a little opening right there called the micropyle. Right, so over here is the pollen and the filament. This is a lily. So what they've done is lilies have a tetrads. These are lily tetrads. These are 4 pollen-producing structures. They've cross-sectioned. This is a cross-section, microsporangium. Anyway, so the sperm out of the pollen tube. Here comes the pollen tube led by the tube nucleus. Turns the corner through the micropyle, and guess what's waiting in there? The female gametophyte. There she is, the female gametophyte. Here comes the male gametophyte. There's the female gametophyte. Now, unique among angiosperms, and something that is very much attributed to their success is what happens next. Something called double fertilization. Remember I said there were 2 nuclei, 2 sperm making their way down the tube. 2 of them, not 1. Right? We normally think 1 egg, 1 sperm, everything's fine, right? And if you look in the female part of it, there is, in the female gametophyte, just 1 egg. There are other cells, there are antipodal cells and polar nuclei that are in the center part of the cell and synergids, but the only egg cell there is just 1, right? All right, double fertilization. Just like we know from before, 1 sperm fertilizes the egg. All right, very good. 1 sperm fertilizes the egg and you get the zygote. Now, and again, this is a lot to say with how these organisms have become so dominant. The other sperm cell unites with the polar nuclei, right, in the middle of the female gametophyte. And notice there are 2 of those polar nuclei. So when that sperm cell unites with the polar nuclei, the sperm cell has N chromosomes. Each polar nuclei has N, because they all came about through mitosis after a megaspore underwent meiosis, back up at the top there. And so what you end up with is getting something that's 3N. Over there on the left, a little bit to the left of all of what I'm talking about, right over here, you notice we got something that is 3N. It's called the endosperm. Y'all remember the other day what I told you endosperm was? In the corn, it's the white part when the corn pops. It's food, right? It's a starter pack. It's a little chuck on the chin to the offspring from the host plant, or I shouldn't say host. That sounds more like parasite, but from the parent plant to the embryo, to the little zygote that's developing there, to give it a little chance to survive. Now, is it going to last forever? Well, no, right? No. I'm stretching it. It's kind of like nursing in a way, right? It's kind of like mammals nursing their young. You don't nurse forever. The offspring have to be weaned at some time, but it provides a little a starter to help you, kind of develop strength and grow and get to the point where you can start taking care of yourself. So that's what the endosperm, in a very broad sense, is doing for that developing embryo, right? So what is this doing overall, though? Upping reproductive rate, right? And if you go back and back and back and back and back to the beginning of the semester, all I've been talking about is differential reproductivity through all of these critters, haven't I? Fittest critters. And this would make you pretty fit. And, remember, in evolution, you don't have to be perfect. So the endosperm didn't make flowers or plants with flowers perfect. It made them have just a little bit more of an advantage than, say, your typical gymnosperm. A lot of gymnosperms still survive, but we're really dominated by these guys, right? Okay, now, as an embryo, depending on what type of flowering plant you are, you develop one or two, as an embryo, cotyledons. That's an important word because it comes back in just a bit. Cotyledon is what we call an embryonic leaf. Think of it as the first leaf an embryo develops. Some plants, the embryo develops one, and then a bunch of others, of course. And then in other ones, the embryonic plant has two cotyledons. Right? That germinates back to where we were at the beginning. Very cool. So big thing here, what we got, right, is this whole idea of double fertilization. That little bit of endosperm, that little bit. Now remember that you didn't find gymnosperms. Gymnosperms don't have that, right? They don't produce that endosperm. And they're going to go more with a little bit of numbers, aren't they? They're going to go more with a little kind of trying to play the probability on that end of it. We'll look at these in the microscope under lab, so we'll talk about that again. But once again, the male gametophyte is on the left-hand side in the pollen grains, and the female gametophyte is inside the embryo sac of those on the right-hand side. All right, I'm going to return back to this idea of double fertilization. Let's follow all those cells again. A little different picture one I found online, but it's a vase. So this is the carpel again, and inside of the carpel there's the ovary, and inside of the ovary there's an ovule, and inside of the ovule there is a diploid cell, and that diploid cell is going to undergo meiosis. That's the megaspore. Undergoes meiosis once, 4 cells, one, right? Only one survives, 3 die, right, or disintegrate. Undergoes mitosis, and there's your 8 cells again. The 2 polar nuclei, the 3 synergids at the top, or sorry, the 2 synergids at the bottom, and then the 3 what we call antipodal cells up at the top. Now there's a microscope picture in the lower right as well. You can see the synergids at the bottom, the zygote, which is a question based on the picture I found. That's going to be obviously the egg cell and the sperm have united. There's the other male nucleus a little bit further to the right. There is the polar nuclei in the middle. They're going to be 3N when that other male nucleus attaches to it. Now, there's a little bit of confusion. Is that the other male nucleus, or is it already fertilized, or not fertilized, but united with those polar nuclei? And then up at the top are the chalazal cells. So what those 8 cells become is 1 egg, 2 synergids. What do the synergids do? The synergids are on either side of the micropyle, and what they do is they tell a pollen tube when to stop growing. So that's that pollen tube that's coming down here, right? When it's its way, those synergid cells, kind of like at the gates, right, say, okay, that's it, pollen tube stop growing, sperm is released, and then the sperm do their job. They do the double fertilization. The 2 polar nuclei we talked about. The 3 antipodal cells, the ones up here at the top, not sure, but I've seen some stuff lately, or I've seen some stuff that also suggests that folks believe that they provide sort of like the anchor that kind of holds the embryo sac inside of the ovule there. Okay. So once these organisms came up with these ideas of flowers and fruits and taking care of young and this kind of dispersal, well, holy cow, everything went crazy. It was, again, like the parents out of town, you're having a party. This is again, a problem for Darwin. He writes back in July of 1879 that during this period when in the fossil record we see this really rapid diversification or appearance of angiosperms, he's like, man, in 100 million years, the rapid development of 100 million years of higher plants, flowering plants, is an abominable mystery. Again, it's the Cambrian explosion of thing. Why did Darwin have a problem? He didn't recognize, again, that there was also cladogenesis, right? Punctuated equilibrium. Periods during evolutionary change in a lineage of organisms that can be periods of very rapid speciation, lots of species. He thought it was just gradualism the whole time, remember? He only knew one mechanism. Some of the earlier angiosperms, Archifructus, about 125 million years ago, but it's probably not the first. Montsechia down in the lower right is one that's been dated perhaps even a little bit older than 125 million years ago. We've seen some numbers back to somewhere close to 200 million years ago. That's what Archifructus looked like in the picture there in the middle. Based on the fossils, we think it had floats, and so based on that, in the upper right is like an artist's interpretation. We think it may have been an aquatic plant. So, you know, it was a flowering plant. Remember I said plants, we were talking about back in chapter 29, some plants went back into the water. So maybe Archifructus was an example of that. Arcta, A-R-C-H-E, always means ancient or early. So this is early fruiting plant is what this is literally saying. We'll always talk about these detective work that they get from looking at fossils. Again, that's how we might have thought it was a aquatic plant, was by the floats. Right, but you can see it's got stamens, so it's got pollinators or pollen-producing structures. It's got carpels, so it's got ability to make fruit. So it's a like I said, it's a fruiting plant. Maybe didn't put a lot into flowers. Maybe some of the earliest angiosperms looked a little bit like this one. This is Ambarella. Ambarella is the closest living relative to what we think the original angiosperms looked like. It's an odd-looking flower on the right. I mean, I'm not sure that would be something that you'd be impressed by if I don't know, say your boyfriend or whatever gave you a dozen. Here, honey, here's a dozen Ambarella. They almost look like something like a candy flower you'd want to eat. They're only found in a small place in the world. Is that the Gilbert Islands or New Caledonia, off northeastern Australia? Australia's this weird continent, land that time forgot. It's been separated for so many years that a lot of very odd critters or old critters or what we call, remember, living fossils are still around. We'll talk more about that when we get into some of the animals that we find in Australia. So maybe this is what some of the earliest angiosperms might have looked like, with not so much in the flowers, but still technically an angiosperm, still a flower. That I would assume these are probably the pollination part of it, maybe. Can't see. The carpels are usually in the middle. All right, we'll go over this in lab too, but among angiosperms, there are some angiosperms that are broken into two big groups. We used to think the groups were phylogenetically sound. In other words, they were monophyletic, but come to find out they're not. I learned the groups as monocots and dicots. Again, that goes back to, as an embryo, do you have one cotyledon, you're a monocot. Do you have two cotyledons, you're a dicot. See, the dicots are paraphyletic, though. So remember what that means. That means it's not something that can be used taxonomically. There are things in the dicots that are not included when we think about dicots. Now they've broken down into something called the eudicots, which means the true dicots. So I have a feeling they've tried to break up that paraphyletic group. That's the bigger group, and then the monocots. Dicots are the trees, shrubs, legumes, flowers, those kinds of things. They use flowers for pollination. Generally, flowers are bigger and more attractive. Monocots are things like grains, grasses, banana, palm trees are monocots. They use wind for pollination in some of them, so they're going to have much smaller flowers. Now, again, this chart on the right we'll go over in lab, but let me run through it once because I think seeing it more than once helps. Monocots and dicots are called that based on how many leaves they have as an embryo. 1 or 2 monocot or dicot. How do we tell if it's a monocot or a dicot? And this can help you when you're identifying plants for your field notebook. Monocots have certain characteristics. For example, versus dicots. Among the venation of the leaves, holding the leaves up to the light and looking at them, monocots tend to have parallel venation. Think about holding up a blade of grass. Think about holding up the part of a palm tree, right? The veins are parallel. Think about sweet gum leaves or maple leaves, how they're more of a net. If they're net, it's a eudicot. If we were to section through the root, or if we were to section through the stem, the monocots, the vascular tissue, the xylem and the phloem, would be scattered throughout, right? Think about if you've ever cut down a palm tree. I have one in lab, I'll show you. Think about what that looks like. Now, in the eudicots, the vascular tissue are in bundles or rings. Now, the familiar counting the rings on the tree, you can do that because you're counting the rings of a eudicot. Those rings aren't lines, they're growth rings of vascular tissue. During the winter, the plants grow less, not as fast. And during the summer, they grow faster. So the little layers get bunched up during the winter, and to our eyes, they look like a line. And during the summer, when the plant is growing faster, the little layers get spread out, and we see the lines more separated. That's the whole basis behind counting the rings on a tree, right? Bunching up the tree, bunching up the vascular tissue. Dicots have a taproot. Eudicots have a taproot, one root with other roots coming off. The pollen is different. And again, if you don't have a microscope with you, that's not very beneficial. One of the other ways to do is counting the floral organs. So counting the number of anthers, counting the number of carpels, the number of petals on the flower. And be careful with that because oftentimes flowers lose petals. Count the sepals, those green flowers underneath. If they're in multiples of 3, it's a monocot. Multiples of 4 or 5, it's a dicot. The one I typically stick with the most is the leaf venation. That's the one I find because oftentimes I'm sitting there trying to count the floral organs and I worry that maybe it's missing one. Maybe an anther fell off. Okay, a little bit more back into the phylogeny of these organisms. The basal angiosperms, there's 3 of those, Amborella, which I already mentioned, which we think is what some of the earliest angiosperms look like. The water lilies, one of which we have here in Florida. This is one I get a lot in the field notebooks. Nymphea odorata, which is the American white water lily. I've seen those on the ponds around campus, actually. The star anise, that's another basal angiosperm. Basal angiosperms, remember, are the angiosperms that are coming off the base group of the ancestral angiosperms. So we would expect this group would be, for lack of a better way, perhaps the most primitive group of angiosperms, okay? So we got the basal angiosperms. The next group is the magnolids, which, of course, is hallmarked by one of the best magnolids, right? The southern magnolia. Great tree. I love these trees. Great smell, wonderful flowers. I love the name, the scientific name of magnolia. Magnolia grandiflora. Isn't that neat? Big flower. About 8,000 species of magnolids. Pepper is a magnolid. Oranges, Persea americana. Right, oranges, those are all magnolids. And then we get the monocots and the eudicots. So again, not all flowering plants are monocots and dicots, and only flowering plants can be monocots and dicots. What it means, you couldn't have a monocot or a dicot pine. Excuse me. You couldn't have it because they're gymnosperms. Only angiosperms can be monocots and dicots, but magnolids, as you see, those are neither one. So. Monocots, about 70,000 species, about a quarter of the flowering plants. For example, palms in the upper right, that's the fan palm. The Florida state tree, which is the cabbage palm, that's another monocot. Again, if you ever cut through a palm tree, you know it doesn't look like the rings that you see on a typical tree. Grass, St. Augustine is a monocot. Orchids are monocots. This is a native Florida orchid. Rose begonia, I think the snake mouth, I think that's a cool name too. We do have native orchids in Florida. All of them protected, by the way. All orchids are protected by Florida statute. That means if you are caught picking or in possession of a native Florida orchid without a permit, then you can be fined and so forth. And then the eudicots, that's the big group, 170,000 species. The legumes, the beans are in this group. The swamp rose, Rosa palustris, that's a Florida, ugly name, beautiful flower. Florida Elysia, Chapmania floridana, this is Florida Elysia. That's a small little yellow flower. I've seen a few on campus. What's really kind of cool about Florida Elysia is it's endemic to Florida. In other words, it's only found in the state of Florida. Sometimes that's why species are protected the way they are. You'll say, why are we protecting these if there's so many in the population? Well, because they're only found in one area of the world. Now, whether we consider Florida Elysia flowers important enough to protect is a different story, but that's what it means to be endemic. Like platypus, right? Speaking of Australia, platypus would be endemic to Australia. Obviously, these flowering plants provide a lot for us, right? We talked about in terms of the gymnosperms, important for resin and that stuff, Taxol and timber. We get a lot of food from from flowering plants, from seed plants, so the wheat, the rice, potatoes, and sweet potatoes. That's about 80% of all calories consumed by humans. We feed a lot of it to our cows. Interestingly enough, we might not be doing that the most efficient way. So you can see that little conversion there. 5 to 7 grams of grain, which a human could eat, can only be turned into 1 kilogram of, or 5 to 7 kilograms of grain, can only be turned into 1 kilogram of beef, right? So there's a little bit of that energy that's lost. Interestingly enough, though, I found out a while back that the #1 consumed meat in the world is not cow. It's actually goat. The #1 consumed livestock meat is goat. I think there's a lot. I've been traveling a lot in the world. I've seen a lot of places, a lot of goat dishes and all that. And I never realized that would have been greater than cow, but apparently it is. Jesse, you're going into medicine too. There's a lot of things that we get out of plants that are important for medicinal purposes, like atropine out of belladonna, which is a pupil dilator, a muscle relaxer, basically quinine, which is good for the prevention of malaria. We talked about Taxol from the yew, which actually is a seed plant, not a flowering plant, but is a seed plant. Curare, which is a muscle relaxant. So lots of things. And many times these sorts of things are better because they don't have quite the side effects. They're more of a holistic nature. So plants are our friends, right? But unfortunately, we're not so necessarily friends to them. Habitat destruction is one of the major causes of plant and really most other species extinction. People say, what's the biggest thing that causes species to go extinct? Is it killing off all of them? Well, not so much as getting rid of their land. You remove their ability to survive, remove their habitat, no matter what organism you're talking about, you imperil that organism. So habitat destruction is a major cause of species extinction, with half of the species suggested or assumed to be extinct within the next few centuries. Not that much time. And a lot of times it's species that don't get a lot of airplay, little insects or grubs of some type that everybody says, what do we care about? But if those insects are important in the ecosystem, sometimes things that we call keystone species are species that kind of hold it in the glue of an ecosystem. And sometimes it's these grubs who are fed upon by these other species that are connected. Remember, that's that meta-evolution thing. So sometimes we cause these species to go in peril just by monkeying with their ecosystem. Every year, about 55,000 square kilometers of rainforest are cleared each year. That's about the size of West Virginia, meaning that in about 200 years, we will have eliminated all of the remaining tropical rainforest. You can see that picture down below, Brazil, between 75 and 2012. So on the right, the purple and the not so green is human development. And this is in the same area, obviously. The problem, as I mentioned the other day, with what we do in tropical deforestation is the land, the soil is not very nutrient rich down there. Remember I said the decomposition rates are usually so high in the tropics that the soil is not very rich. Most of the nutrients in the tropics are in the living thing. Things decompose very quick, and there's lots of things down there growing that sucks all those growth nutrients up. And then what do people do when the land is no longer farmable because it's not fertile? They cut down more forest and keep going and keep going. Now, of course, the loss of forest from the species perspective, but the loss of forest from the global perspective is a problem too, right? It's less carbon dioxide that's being taken up by those trees during photosynthesis, which carbon dioxide is one of the implications for global climate change. It's one of those gases we know as the greenhouse gases has been implicated in causing specifically temperature changes. Global climate change is more than just the temperature. A lot of people focus on this whole idea of global warming. It's not necessarily just warming. It's a change in the typical temperatures in there. It's a shifting of the seasonal patterns. This last slide, I'm not going to spend a lot of time going over, really just to kind of nothing more to orient you. This is kind of a little review slide to look at the differences among the major groups of plants that we've been through, the mosses or the bryophytes. Remember, not all bryophytes are mosses. The ferns, what do we call those? The manilophytes or the pterophytes. The conifers, specifically a gymnosperm and then the angiosperms. And then I just kind of list some of the features. So this is kind of a, I don't know, call it a learning aid, maybe a way to organize your thoughts about the differences between these organisms. Okay, folks, that's it for Chapter 30. I want to do Chapter 31 now. I want to get into the fungi. I just noticed I don't have that chapter loaded, so give me 1 second while that loads. Anybody have any questions for me? Any of you two have any questions for me based on the stuff I've gone through as far as that goes? This will load pretty quick, so I'll be ready to do this in just a second. What time is it? Oh, good. So is it Chapter 31? Yeah. Chapter 31, which is the fungus, I sometimes have to actually not do that chapter, and I feel bad about that because the fungus are really neat critters, but I get kind of pressed for time in the semester, so sometimes I cut out this chapter and lecture, but I'm able to do it this semester. I'm glad we're actually able to do the lab on it, which I'm really glad about because it's a neat lab. But, and I'm looking, and that's correct, I do not include this chapter in any of the lecture exams, so you don't need to know the fungus chapter 31 for any of the lecture exams. So the next one's 27 and 28, the one you take Monday, then the one after is 29 and 30, and then the last one is 32. But you do need to know the fungus as far as for the plants, okay? So as far as exercise 6, which is the fungus exercise, not the one we'll do Monday because we're finishing up plants on Monday, but the one we're just about ready for the fungus that we'll do on Tuesday. Go. Fungi. Neat little critters. Especially I feel bad when I have to cut them out, but sometimes because of time I do. And I guess I do because I can't really cut out any of the plant chapters because there's 2 of those and you got to go all through all that. And the animals are the same way. But there's only one chapter on the fungi. And that doesn't mean they're not important. but it means that they're very different than the other two groups of organisms, the big groups of organisms we've talked about, the plants and the animals. Of course, they're substantially different than the protozoans and the prokaryotes. But one of the things about the fungi that I think always fascinated me is the diversity of form. They can grow in any different form. They're not constrained to any particular color. There can be this one. This is, look at the name of this one, Lactarius indigo. Blue color. These grow in Florida. I've seen Lactarius mushrooms on campus before, and they have a kind of a blue or a bluish gray tint to them. Some of them can grow, their typical toadstool scents. Some of them grow like these Lactarius, sort of like a fungus. Neat painting too, isn't it? Because look over here, the guy, there's spores in fungi too. So look, he's drawing the spores over here. He or she, whoever did this. That stuff that you see growing on the trees here in Florida, the bracket fungus that can grow in that form. So there's a lot of different ways that fungi can live. So they're really not plant-like, and they're really not animal-like. They're really like a real different way of going about life. And another really sort of interesting little fact about them, I get this all the time. What's the world's largest individual organism of what species? Blue whale or redwood trees, and those are pretty good size, right? But it's actually a fungus. There's actually a fungus. It's in Oregon, I believe, that is of this species, the honey mushrooms. These are honey mushrooms in the background. You might have seen these in Florida. Honey mushrooms, Armillaria ostiae, are a very common species of mushrooms. There's nothing unique about these mushrooms. But there's one of them, one individual, and they've done this through genetic testing. And as I get in more on this, you'll see how they're able to figure this out. Out in, I believe it's a forest in Oregon. Maybe there's another one near Spain or somewhere around there. One individual that covers 9 million square meters of forest. It's over 1,900 years old. It weighs hundreds of tons. It's on the order of somewhere 3.5 miles across. So when you tell people that, well, actually, it's a mushroom growing in the Oregon forest, they're usually pretty flabbergasted. A mushroom that's 3.5 miles across. Now, if you're thinking there's this huge toadstool that's 3.5 miles across, well, today we'll get into the basic structure of fungi, and you'll learn that when we realize where fungus or how fungus spend most of their time, you can see how an organism could grow that large. Pretty speciose group of critters, about 1.5 million different fungal species, growing either single-celled, yeasts, for example, or fungus. They're single-celled, or the typical multicellular type. They reproduce sexually. Some species more inclined to reproduce asexually. The basis, and speaking of sexuality or sexual reproduction, the basis of how these organisms are classified, the large groups, the phyla of the different kinds of fungi that we're going to go through, is based on the way they reproduce. All right. Now, plants produce food through photosynthesis, and they go through respiration, of course. We, of course, go through respiration, but we need energy to do that. So we eat plants, or eat things that eat plants, eat meat, or whatever the case may be. And we bring the food inside of our bodies where we digest it, in our stomachs. All right, nothing new there. Fungi do something a little bit different. They also have enzymes that help them digest food, but these, in this case, they're called exozymes. Exozymes, because the way fungi do this is they secrete these exozymes into the environment around them. And so they're digesting the nutrients, and then they simply absorb those nutrients from their surroundings. So you see, they're doing something we call extracellular digestion. They're digesting the material on the outside of their bodies, on the outside of their cells, and then basically using diffusion and other uptake methods by which to take in those nutrients. So that's a little different, right? So if you think about, in contrast, the way we do it, we would engage then in intracellular digestion, right? We do the digestions inside of our bodies and inside of our cells. We bring the food into our cells and begin to break it down. I know there's the digestion, of course, it starts in your mouth and in your stomach, but the digestion process occurs inside of the organism. Weird group then, right? more closely related to animals, more closely related to plants. Fungus, come to find out, are actually share some characteristics, some relatively basic characteristics between animals and a fungal ancestor, somewhere around 460 or 670 million years ago. We'll talk about those a little bit more as we start getting into some of the properties of fungus. So your closest living relative outside of the animals would not be a plant. I don't know if that makes it any better or worse, but actually a fungus. As I mentioned, the exozymes. Now, back to that idea of that 3.5 mile across fungus, it has to do with something known as the mycelium. Now, the mycelium, if you will, just for right now, just for these next couple slides, we'll call the mycelium the roots of the fungus. They're not really roots, of course, but they're the tendrils or the devices, the structures that the fungus use to penetrate down into what they grow on. See, fungi are decomposers, right? They can break down things like cellulose. They can break down stuff like lignin. Lignin is that stuff in the lower left that there's the atomic or the chemical formula or the structure for it. Lignin is that stuff that makes trees really, really hard, and it's a very difficult organic material to break apart, to digest. There's only a few organisms can do it. One of them was prokaryotes, right? We talked about that, and fungi. And that's real important because all those little circles in there, you remember from Bio 1, and you can even see, in some of the symbols, a lot of carbon in there. You want to get that back into the environment. So nutritionally, these fungus are real important ecosystem-wise because they're good in terms of decomposition. They can break down these things. And animals, of course, we're beholden to that, right? We can't break down cellulose. We can't break down lignin. So we depend on other organisms in the environment to do that for us, to break that stuff down. Now, there are a few exceptions, right? Kind of exceptions, technical exceptions. Cows can digest cellulose, but remember, cows can only digest that because in their stomachs there are a symbiotic bacteria. They don't do that themselves. They do that in conjunction with other organisms that live inside their gut. And there are some fungi that are even carnivorous, believe it or not. So be thinking about that next time you stroll up to the salad bar Ruby Tuesdays about the fungus that are sitting there. Some of them might be carnivorous. Body structure. Another weird thing about these critters, I've heard it said, and maybe this is the best way to think about it, in fungus, the focus of reproduction is not on the cell. In other words, it's not the cell that's really necessarily important in terms of reproduction. It's the nucleus. It's the genetic material, which really is all what cellular reproduction is about, right? You got to have karyokinesis, then you divide the cell. Well, fungi have, in many cases, have either delayed or kind of skipped or modified the whole cytokinesis part, right? Fungi grow as long, slender filaments, and these filaments can either be a cell, or many cells, right? And so this kind of really challenges our idea of what's called multicellular, because fungus are considered multicellular, but in some cases, these long filaments, these things called hyphae, those are the little roots that I was talking about, the tendrils. See in the picture in the yellow down below, the little projections of the fungus. If you ever seen black bread mold growing on the bread, that's what all those little tendrils are. Those are hyphae. Those long little things can be made up of a few cells or even just a cell, and we're still talking multicellular fungus, right? So I don't have a great answer for you there, but it's just kind of keeping in mind that we're focusing on the idea that in fungi, this multicellular term refers to the fact that really it's multinuclear, right? Now, some of these hyphae can be synocytic. That means they're continuous, like the one on the right. It's one big giant open with look at that, polynuclei. Right? Many different nuclei. Look at that. There's a bunch of nuclei in that one gigantic cell. Others can be septate. Now, it doesn't mean that's different cells. It's one cell with these divisions in between, these septum that allow materials to flow through. The reason why hyphae would be advantageous, all right? The reason why having these one continuous pipes, or in some cases those with continuous pipe but maybe some septate in between, is this allows for very rapid expansion During times when fungi want to reproduce, and fungi are going to reproduce in some cases when the environment is deteriorating. So fungi are going to reproduce when it's time to think about making a lot of offspring very quick because the environment is changing. Now, hopefully, you go back to the beginning of the semester. If the environment is changing, the only way that organisms can hope to survive the future environment is to, by chance, genetic recombination, mutation, whatever you want to throw in the mix there, by chance they've developed a favorable a phenotype that is the most fit. So when the environment is changing, many species of organisms, what they're going to do is reproduce at that time, because that's when you get a lot of variability. Because during reproduction, you undergo meiosis, and meiosis generates variability. So what's happening with fungi is these hyphae, these long continuous cell or just a few cells, allows during time when reproduction is necessary for cytoplasm to very quickly grow through the flow through these tubes and cause these hyphae to swell. All right, and what they do is when they swell, that's when, in many cases, depending on the species, that's when you see them. You see, the mycelial, the thing I mentioned before in the red, the mycelial is underground. If you dig into the dirt a little bit, like you see on the right-hand side, all those little white stringy things that are connecting all these decomposing parts of plants, this decomposing organic material, under a microscope, you would notice what those are. Those are hyphae. And mycelial is a mat of hyphae. And hyphae are those filament of cell or cells. They're growing underground. Now you know why that big honey mushroom is not a big 3.5 mile, toadstool. It's 3.5 miles from one side of the hyphae of one individual. They test this genetically, of course, to the extension of the hyphae on the other side. So it's all underground. So, and I got a picture of this. That's why sometimes mushrooms seem to appear out of nowhere, right? It rains the night before and you got in your yard, boop, there they all are. Well, they were there. The mycelial mat underground was extending through the subterranean, secreting exozymes, right? Secreting material to digest food. And then when rain came, and as I said, in some species, it's when the environment deteriorates. In some species, it's when the environment is conducive. During the rain, things are going to be wet. Good time for fungus to germinate. So some species go, oh, it's time to produce offspring because there's resources, there's moisture out there for those spores to survive. Those toadstools, those things that pop up to the surface, they're spore-producing structures. That's their job. We're going to look at a toadstool lab. Underneath the gills of a mushroom, all of those are lined with spore-producing structures. You're going to make lots of spores very quickly, and you're going to pop up out of ground to do that. But most of the time, you're going to be spending your time underground as this mass of connected hyphae, either synocytic or septate. There's some cool pictures down below. You can see the septum in the septate. You're going to grow through the subterranean or into the bark of the tree or even on the surface of the tree. You don't have to be underground. And you're going to grow these little tendrils, which is massive amounts of surface area. Huge surface area for lots of absorption of nutrients. And that's in keeping with their lifestyle, right? Exozymes. They're growing in this digestive soup of digested material, so they're going to want a lot of surface area to suck all that up. Oh, one of the things that connects us with fungi at a very basic level in terms of ancestry is the presence of chitin. In the cell walls of fungi, there is chitin. Chitin is that, well, it's a modified carbohydrate. It's the stuff that makes the exoskeleton like some arthropods, like in that male rhinoceros beetle in the picture. It's the shell material on on insects. And as I said, the cell walls of fungi also have chitin, so there's a connection there, right? A distant connection, but nonetheless a connection. You don't find chitin in plant cells. Oh, and as I said, there are carnivorous ones. This is kind of neat. Nematodes are little worms. We'll actually dissect a nematode. Very simple worms, microscopic although the one we're going to dissect is a little bit bigger. This one is microscopic, but very often found in soil. Farmers talk about nematodes. Nematodes are important in many cases, certain species, for soil richness. But nematodes are food, right, for some species, and some species of fungus can develop these hyphal loops that can spring around and wrap up these nematodes really quick. The loop can close in something less than a second, so that's moving pretty doggone fast for a fungus. So the poor little nematode gets a little bit too close, and these hyphae shoot out and wrap around, and then what they would do is just continue to wrap hyphae around the nematode and then begin to digest it. Plants aren't safe either. There are fungal hyphae that can produce these haustoria, or singular haustorium, which the little fingers are projections of the fungal hyphae inside of the plant cell, and they can in some cases they can parasitize the plant cell. There are also species that don't parasitize the plant cell, though, that actually provide benefit to the plant cell. It's kind of that old adage of one person's poison, another person's food. So fungal hyphae can be used parasitically with Haustorium to extract and use the plant cell as a host, and that would be a parasite, while other fungi can actually produce Haustorium to benefit not only themselves, but the plant cell. I mentioned a while back when we talked about the evolution of life on Earth, the idea of plants pollinating land and doing that in conjunction with a fungus, and we called it mycorrhizae at the time. Mycorrhizae on the right-hand side in the upper picture, those are little plant roots, and little hairy things all over it are fungal hyphae, mycorrhizae. Mycorrhizae are in a mutualistic right, relationship with plant roots. Very important. So important that, like I said, 90% of all vascular plants, right? So we're going from the mosses, from the ferns forward, vascular plants have mycorrhizae on their roots. If you take two plants side by side, see the pictures up above, you spray the one on the left with fungicide, something that would kill a fungus, something that would kill mycorrhizae, and let them grow otherwise the same, you can see the obvious difference in plant growth. The one on the right-hand side is a much more vigorously growing plant with those mycorrhizae. It's mutualistic. The mycorrhizae extend out into the soil around the plant roots, so they extend the root system, they increase the surface area, they increase contact with the soil, they're secreting these exozymes. These plant roots are growing in amongst these tendrils, these hyphae of mycorrhizae, who are turning the soil into this nutrient-rich soup. Right? It's kind of like, it's ABC food, already been chewed. It's already been digested. So, of course, the mycorrhizae, the fungus, is doing this, digesting these materials for itself, right? It's not doing it for the benefit of the plant, but it's going to digest more materials than it itself can absorb. So the plant absorbs the rest. And in mutualistic, the plant actually does provide things for the fungus. It provides one thing it provides is carbon, that very important structural element for living things, right? Carbon-based life forms. Speaking of things that grow on things, and they're not up here on campus, that's too bad, but every once in a while, depending on the conditions, I'll find these Indian pipe. These are flowering plants. That's a flowering plant in that picture, that white thing. Not the green, but the white thing is Indian pipe, Monotropa uniflora. This is a flowering plant, does not photosynthesize, right? It's white. Got to be green to photosynthesize. No trick here like with the red algae at all. This thing does not photosynthesize. It's a non-photosynthetic plant. I don't know if you knew that those existed or that we even had these. Like I said, I've seen them growing on campus. This is a really cool example of something called a tripartite relationship. See there in the red? A relationship of three organisms living together. Monotropa uniflora, the Indian pipe plant there, the roots of that plant grow on the fungus, the mycorrhizae, that grow on the roots of plants. right? So many times you'll find Indian pipe growing underneath like oak trees. So the oak tree's roots that are growing out have mycorrhizae growing on them, and the Indian pipe roots are growing in amongst the mycorrhizae that are growing on the roots. So it's the plant roots, the mycorrhizae, the roots of the Indian pipe growing together. And so symbiotic relationships can be more than just, plus and plus, plus and minus. It can be plus, plus, and plus, or whatever other mixture. So that mega evolution, right, at that much bigger level, critters all living together. My German teacher, I remember in junior high, Arbeiten zusammen. She do the gears, work together, work together. She wanted us to work together. It's the only German I remember. So all these critters are Arbeiten zusammen. They're all working together. So mycorrhizae can grow either as ectomycorrhizae, these grow around the roots between the cells, or they can be mycorrhizae that are called arpuscular, which again form these arpuscules or these hostoriums, see in the picture on the right, where it penetrates through the cell wall but not into the cell membrane. In the picture on the left, and we're going to look at this in lab, this is the plant root right here. The blue covering on the outside, that's the mycorrhizae. In this picture, it's the green hairy things. In this picture, it's the fur around the plant root. That's how extensive. So it makes a covering of these hyphae around the. So you can see the vascular tissue. You can see the xylem and the phloem of the plants there. So that's pretty obvious. All right, reproduction in these guys, it's a little bit weird, right? We have haploid stages, we have diploid stages, all right? We have something called the heterokaryotic stage, okay? And we have something called plasmogamy and karyogamy. Now, plasmogamy and karyogamy are nothing new, and I've defined, or the book has defined them. Plasmogamy is when the cytoplasm of cells fuse together. And then karyogamy is when nuclei fuse together. So what I mean by nothing new, if you think about egg and sperm, when fertilization occurs, the first thing that happens is the cells fuse together, so plasmogamy, and then the nuclei fuse together to form the zygote. What's different here about fungi, though, is the time between those two things. From plasmogamy, when the cells fuse, to karyogamy could take hours, depending on the species, days or centuries. So the purple part of the arrow there, that's what's really different about fungi. Fungi can spend an extended period of time, up to centuries, in something called a heterokaryotic stage, in a stage in which a cell or many cells, right, actually exist as having nuclei that are unfused within a cell, a heterokaryotic stage. And this extends to being more than just a couple of nuclei. It can be many nuclei like we saw in the picture. Now, the advantages of being heterokaryotic, the advantages are similar to being diploid. All right. When you're haploid, you only have one set of genetic information. Okay, and that works in some applications among some critters. That's what gametophytes are in mosses. When you're diploid, you have two sets of genetic information. And remember, we talked about in evolution, having two sets means that recessive allele can potentially be beneficial. And you can have differences in genotypes. Homozygous dominant, heterozygotes, homozygous recessives. Right? So you can have some genetic variation in there. So being dikaryotic or multikaryotic in some of these, depending on whether there's two haploid nuclei or several, is similar to having the advantage of being diploid. Now, is it the same as being diploid? No. Is it better or worse than diploid? Irrelevant. Irrelevant because for us, diploid works, but for fungi, being heterokaryotic works. If it didn't work, they would not be here, right? Remember, let's not be Aristotelian and go back to this idea of things are on a ladder. It's not a ladder. You know, organisms can have many different ways of surviving just as long as you're fit, just as long as you have that advantage in the environment. All right, so now when karyogamy does occur, then we're kind of back into something that we're very familiar with. You get the zygote, but they don't last very long. Again, it's not the cell. it's really not the focus of reproduction. It's all about the nucleus, because that zygote very quickly, that's the only diploid stage for a little bit of time, quickly undergoes meiosis. And I put question mark purpose there, right? Because again, always remember, meiosis, that's variation. And we produce spores that are different from one another. These are called sexual spores. In fungus, we oftentimes will go, we have sexual spores, and we have spores that are also produced asexually. Because on the left-hand side, you can see you can also have asexual reproduction. Very simple. Spore-producing structures germinate, and either asexual or sexual spores produce mycelium. They germinate, and they form those long chains of cell or cells. Now, if you go into sexual reproduction, it's not a matter of, you don't have male and female fungi, but you have what we call mating types, okay? And those mating types attract one another through chemical means, like pheromones. Pheromones are basically, I always tell folks, pheromones are like environmental hormones. If I could secrete a chemical into the environment that would signal something, then those would be pheromones. So different mycelia of different fungi, different mating types, different genetics, right? Different individuals could use these pheromones to attract one another and to prevent breeding with members that they're closely related to, okay? So if you want to really kind of think about compare and contrast reproductive strategies, these guys with everybody else, it's this process right here, this heterokaryote, the part in the purple that's really unique. And the reason why they're doing this is because, like I said, you get the advantages of being diploid when you're heterokaryotic, but you still are retaining this weird idea of what a cell is. A cell is one big thing with many nuclei, or maybe just a few things with many nuclei. Of course, asexual reproduction, like I said, is very often seen. You know, many engage in both sexual and asexual. Some are limited to just one type. There are a few species, I don't know how much of this has changed, about 20,000 species in which only asexual reproduction is known. In other words, what we mean is there are some species of fungi out there in which we have only been able to observe how they reproduce asexually, which is nothing more than spores germinating back to mycelium, making other spores. Remember I said at the beginning, the way fungi are classified is they're classified based on how, in the bigger groups, how they sexually reproduce, what structure they make. And we'll get into what I mean by structure in a bit here. But if that structure has never been observed in our observations of a fungus, then historically what has been done is there's a group called the deuteromycetes, which literally, or has been also called the imperfect fungi. Not that there's anything wrong with them, but what we mean by that is this is kind of a collect-all group for fungus that we're not sure what group to put them in yet, right? And maybe this will make a little more sense as we get into the groups. But like I said, the different groups of fungi, phyla, right, are placed there based on the type of reproductive structure they make when they sexually reproduce. And if we've never observed a species engaging in sexual reproduction, right, then we don't know what group to put in, so we put it in deuteromycots. Molds are very well known for asexual reproducers, right? They have these very filamentous fungi like that orange, that's penicillium mold that's growing on that. What you see in the picture there, these are the spore-producing structures in the penicillium. These are all can produce spores. Penicillium is really kind of neat under the microscope. Yeast, I mentioned, that's another type of fungi very well known for asexual reproduction, reproduced by budding, where a parent cell simply divides. Asexual reproduction, right? No genetic variability, so you're going to do a lot for survival in terms of what the prokaryotes do. You're going to produce lots of offspring. This is how yeast, scientific name for one of them, for yeast, Saccharomyces cerevisiae, right? So reflecting its roots in distilling, right? That's what you use to produce alcohol, is yeast. Yeast and anaerobic respiration, right? Turn sugar into alcohol. Fungi in the fossil record somewhere about 460 million years ago, but that probably has a lot to do with their inability to preserve very well. The molecular clock tells us that animals and fungi diverged from one another somewhere around 1 billion or maybe even 1.5 billion years ago, so very early on in the Earth's history. The ancestor was something like that critter on the right, aquatic, flagellated, probably unicellular. But as I mentioned before, animals and fungi are closely related, relatively closely related. We form a group of these posterior flagella critters. Remember, I call them the opisthocons. Opisto being rear or posterior, and cont being a flagella. But here we go again with protozoans, right? Fungi, if you look at the chart below, fungi are actually more closely related to a group of protozoans known as nuclarids. Nuclarids, I found a picture. There's nuclarids on the, no, those aren't nuclarids. Those are just an example, again, of opisthoconts. Those are sperm cells. But nuclarids are protozoans. And again, just like protozoans being mixed in everything else, they are mixed in with the fungi as well. So fungi are more closely related to a protozoan than they are to other groups of critters that aren't protozoans, that big paraphyletic group. Among fungi, we have the chytrids, a small group of about 1,000. Then we have a zygomycetes, another very small group, the glomeromycetes, and then two really big groups, the ascomycetes and the basidiomycetes, with 30,000 species. The ascomycetes being the biggest group with 65,000 species of fungi. So let's start with the chytrids first. About 1,000 species. Ubiquitous means they're found everywhere. Whenever we talk about an organism being ubiquitous, it means that it's humans on Earth would be ubiquitous. We're found everywhere. Flies, mosquitoes would be ubiquitous. Chytrids have also been found in some very bizarre places, like around those hydrothermal vents that we talked about. Remember where it's really can be really hot with a lot of toxic fumes, but there are critters that make a living there, and some of them are members of this phyla, this Chytridiomycota. They act as decomposers. They can be parasites. That's the picture on the right. This is a chytrid fungus that's attacking a Odygonium. Odygonium is an algae, kind of like Oscillatoria that we looked at, and see that chytrid is actually burrowing, right, its way into the Odygonium, and will digest the cellular contents. Life is rough, man, even at a cellular level. They can be mutualists, right? Plus and plus. These organisms diverged from other fungus really early on, and they have as being opisthocons, they have flagellated spores. Yeah, we talked about this before. Spores can either be flagellated or not, and when they're flagellated, we call them zoospores. Zoo is a word or a root we typically reserve for just referring to animals. But as we learned, one of the biggest characteristics about animals is their ability to move. So things that can move, we sometimes will put that zoo. It's not zoo because it's zoology, not zoology. We'll put that zoo part into it so that we're reflecting a spore that has a flagellum. This is unique to this group, these flagellated flagella. You probably have a lot more experience with this group, though. I mean by experience, if you look in the upper right, this is what causes the fuzz that grow on your strawberries or bread. This is what we typically call black bread mold, or the zygomycots. I said, especially among these groups that we're going to go through, it's all about reproductive, how you reproduce. So with that in mind, let's start in terms of reproduction, looking at this chart over on the purple side on the right. See, right after plasmogamy, there's that extended heterokaryotic stage. Zygomycots or zygomycetes are named because during this time, two mating types, again, those are the blue and the red, those are the hyphae of two mating types, come together and formed something called a zygosporangium. Initially, when they touch, it's a gametangia, so I'm kind of backing up a little bit, sorry. Then a zygosporangium, and that zygosporangium, that's where the name comes from, grows into this metabolically inactive, but this really resistant-looking thing but it's lumpy. We're going to look at them under a microscope, so it looks like it's tough. It's resistant to freezing. It's resistant to drying out. It's resisting to the environment deteriorating. You see, black bread mold are more than happy to asexually reproduce. Notice I wrote in red, that's typical. Producing asexual spores, just going crazy, growing all over your piece of bread, right, or growing all over your strawberry. But when the environment deteriorates, they're going to form these zygosporangium. And since heterokaryotic can be a long stage, they can stay in this it's kind of like having a seed, folks, or a spore or a cyst. It's something that can remain and wait for the environment to get better. And then when the environment gets better, if it does, then karyogamy can occur. And we get this very short-lived zygote, right? This short-lived zygote that is multinucleate. Each one of those reds and blue dots, when the gamma-tangia, when plasmogamy were occurring, those are nuclei. So that diploid nucleide then is going to turn around very quickly and undergo meiosis. Bam, we get variability. And now we get spores. So these little lollipop things, these sporangia, similar terms like we saw in plants, are going to produce spores. But now these are going to be sexually reproduced spores as opposed to the ones asexually, right? And they'll go out and find some other place to hopefully another piece of bread or something like that. You can see that rhizopus is a real common black bread mold. That's the one we'll look at under a microscope. You can see it there where it is growing on the bread. And hopefully if these spores find a place to germinate after they disperse, then they grow and become different mating types again. And mating type is just different different individuals. So zygomycetes can be molds, it can be parasites, commensals. Commensals are different than mutualistics. Commensals are one gains a benefit, and the other organism is neither harmed nor helped, kind of like the egrets and the cattle that we talked about. Zygomycetes it can grow very quickly. They have synocytic hyphae. So hyphae have no septate at all, so they can quickly flood the edges of those hyphae with cytoplasm to quickly spread over your piece of bread or your strawberry. In the lower or on the left-hand side, that's Pilobus. The one with the weird looking, those are little sporangia that can actually, what they do is they actually can turn in a direction that is more conducive for spore germination. And off the top of my head, I can't remember how they sense that. I looked it up one time and it was kind of neat. But these sporangia, kind of like the elater pushing the horse stem, the spores out, these sporangia can actually point in a direction where they, based on environmental conditions, where they think the spores have a better chance of surviving. It's kind of freaky. Can't remember exactly what the cue was, but it was neat. This little group, the Glomeromycetes, only about 150, 160 species, small, at one time actually lumped in with the black bread molds, but really, really important. Nearly all Glomeromycetes form the one type of mycorrhizae, the arpuscular type, right? The mycorrhizae, again, are the tendrils around the plant roots. And the arpuscular type, see on the right, that's a really cool picture I found. These are the arpuscules, or the arpuscule, the little extensions of the hyphae growing into the plant cells. Remember, these this could be Haustorium in a parasitic fungus, but in mycorrhizae it would be a mutualistic fungus. So they're providing it's just a matter of balance. As long as you're helping each other out, then you're mutualistic. If one is hurting and one's helping, then that's obviously a parasite. Sac fungi, Ascomycota, Ascomycetes. Again, it's all about how these things reproduce. So down in the lower left, I don't know what species this is, but this is your typical sac fungus. That red bowl thing is the fruiting body, is the reproductive structure. Ascomycots, 65,000 species, 75% of the fungi. Include some of the bread yeast fit in this group, and molds. Morels, Morchella, this one over here, this is a sac fungus. If you think, wow, that looks really different than the one on the lower left, think of it as a bunch of little sacs. See, those little pits are sacs. Truffles, that's a truffle, tuber melanosporum, or truffles. Truffles are those really expensive fungus. I found online, if you look over on the right, you can buy, what does that say, flash frozen black winter truffles at $575 Well, $573 for half a pound. That's amazing to me. I've never had truffles, but from whatever, they must be just so incredible to spend that kind of money. Truffles are the things that you remember that they use female pigs to find. Truffles grow underneath leaf litter, so the female pigs can actually sniff them out. And before the female pig gets a chance to devour them, then the farmer or the fellow right behind him picks up the mushrooms. The reason why female pigs are so good at finding them is because female pigs is because truffles give off an odor similar to male pheromones, male pig pheromones. Lichens, like you see on the right-hand side, are that symbiotic relationship between something that photosynthesizes, like a green algae, or maybe a prokaryote and a fungus. Many times, or often, about a quarter of the fungi that are found in lichens are going to be of this group, of these ascomycetes, of these sac fungi. So that's all I got time for now. We'll continue on with this tomorrow, talking about the reproductive strategies, but I'll go ahead and finish lecture on fungus right now. Let me finish the recording.